Good morning, everyone. Thank you for joining us today. We will start in a few minutes just to give time for everyone to have a chance to join the webinar. Hello, everyone. Thank you for taking the time to join us today for this exciting webinar being brought to you by Cintiqa. My name is Ivy, and I will be your host for today's webinar. Our webinar today is entitled Advancements in Intravital Microscopy, Insights for Preclinical Research Applications. Welcome to today's webinar featuring Dr. Abhi Karunendran, an applications specialist here at Cintiqa. Dr. Karunendran holds a master's and a PhD from the University of Toronto in cell and systems biology, with research spanning multiple photon microscopy and cell biology. Following her academic journey, she completed a postdoctoral fellowship at Harvard Medical School, focusing on intravital microscopy. In her role, Dr. Karunendran is dedicated to connecting with the researchers and highlighting the utility of intravital microscopy in various research applications. Today, she will discuss how this technique can support research efforts. Before we begin, I'd like to briefly mention a few housekeeping items. We anticipate the presentation will be about 45 minutes, which should leave us with some time for questions at the end. Please submit your questions in the Q&A chat box throughout the presentation, and we will go through each question at the end. We will also create a transcript of the questions we do answer during the session, as well as those that we may not have a chance to address. This transcript will be made available afterwards. Please also note that we are recording today's session. So if for some reason you lose connection or you can't hear anything clearly, you will have access to the video file to review later. Before I hand over the presentation to Dr. Karunendran, we're going to kick off this webinar with a quick poll to gauge where everyone stands. So we have a poll question for you. Our first question is, do you have prior experience with using in vivo imaging? Yes or no? Please answer, and I'll give you a few seconds to answer before we can delve into our presentation. Great, thank you all for participating. And without further ado, I am pleased to turn this over to Dr. Karunendran for today's webinar.
Thank you, Ivy, for the lovely introduction. My name is Abby, and I will be going through the advantages of intravital microscopy and how it could be utilized in preclinical research. So intravital microscopy is a powerful imaging modality that allows for real-time visualization of biological processes within living organisms. It stands apart from other imaging modalities such as X-ray, ultrasound, CT, While these modalities do offer valuable insights, they tend to provide static snapshots of biological processes at a macroscopic scale and with limited resolution. In contrast, intravital microscopy can offer dynamic insights into biological processes in vivo. Why is this crucial? Well, because intravital imaging would accurately capture complex physiological phenomena within the living organism, whether it's to study drug responses, immune system interactions, or disease progression. This modality would allow researchers to observe these processes in real time and within the context of a living organism. So in order to have a successful intravital imaging session, there are several key principles that are required in order to be able to observe these biological processes in detail and precision. First being, you require optical access and tissue penetration. Intravital microscopy relies on an advanced optical system and imaging technologies in order to be able to achieve this, to be able to penetrate the tissue for visualizations of cellular structures deep within the living organisms. This would include utilizing specialized objectives with high numerical apertures, which could be used to collect light emitted or scattered from fluorescent labels within the tissues in order to maximize signal detection and resolution. This could also mean using multiphoton excitation, which would utilize a longer wavelength light source in order to penetrate deeper into the tissues while minimizing light scattering and phototoxicity, and allows for imaging at greater depths. And this would also require surgical procedures that could expose the tissue under the objective. Second is you want to minimize the amount of damage that is done and to maintain physiological conditions. And for this, you would need to employ several techniques. Again, you would need gentle sample preparation, so the animals have to be typically anesthetized and prepared for surgical procedures in order to expose the tissue of interest while also minimizing trauma. Careful handling and positioning of the animal is all essential in order to prevent tissue damage. You also have to consider the light exposure. So light exposure to excessive light can also induce phototoxicity and photobleaching, which would lead to tissue damage and compromised image quality. And so imaging parameters such as laser power, exposure time, all have to be carefully optimized in order to minimize these phototoxic effects, while still obtaining a sufficient signal intensity. In addition to that, we need to maintain the physiological condition of both the tissue and the animal over a long period of time. This would include temperature and oxygenation control in certain scenarios in order to preserve tissue viability and function. This also includes microenvironment control, so techniques such as perfusion of tissues with physiological buffers and imaging media may be required in order to keep the tissue hydrated, keep it balanced, and ensures the preservation of normal cellular functions during your experiment. And lastly, you would need to be able to capture these dynamic biological processes over a period of time, and you need to be able to use this in order to study real-time events within living organisms. This includes high-speed imaging. So advanced imaging systems need to be equipped with high-speed scanners and detectors in order to be able to acquire sequential images at a rapid frame rate. This would allow researchers to capture dynamic processes with temporal resolution ranging from milliseconds to minutes. This also works in the long run when you look at longitudinal studies. So time-lapse imaging would facilitate longitudinal studies, and this would enable researchers to track changes in cellular behavior, tissue morphology, and physiological parameters over extended periods. This is required. You would need sophisticated imaging tools and softwares in order to be able to analyze time-lapse imaging sequences and extract quantitative data to visualize these dynamic events. All the things that I've just mentioned is usually something that you would have to integrate yourself through trial and error into a conventional scope, which would be time consuming and a lot of work. But with IVIM Tech, they have meticulously designed a system that advances the imaging technology for intravital imaging, but also prioritize user experience. So I would like to introduce the IVIM system, which is a all-in-one box solution that seamlessly integrates all the essential features that you would require for imaging at high speed and resolution. The important factor of this design is that it's a compact system and it's been meticulously engineered to ensure maximum efficiency for researchers. And therefore, they can delve into their experimental interviews without needing for manual adjustments, thanks to its hand-free capabilities. It also comes integrated with various apparatuses for maintaining stable physiological conditions during the imaging sessions, which I will dive into in a few slides. So just a little bit about the IVIM system. There are 5 product lines that are provided with the system, depending on which modality you would want to use in your experiments. So there's confocal microscopy, the IVMC, which is listed as number 3. Confocal microscopy is a powerful approach for in vivo imaging because it gives you information in 4 dimensions, X, Y, Z, and time. And because it utilizes a laser light and a pinhole mechanism, it allows precise focusing on a specific point while deflecting and reflecting any scattered light in order to increase signal contrast. There's also the 2-photon modality, which combines the flexibility of a traditional microscope with the high-resolution imaging capabilities of multi-photon microscopy. This allows you to be able to penetrate the tissue at greater depths and also gives you the capability of second harmonic imaging, which allows you to do some label-free imaging of fibrous tissue. And for this, you have 2 separate 2-photon modalities. You have the IVIM-MS listed as number 1, which is 2-photon at a fixed wavelength of 920 nanometers. And then there's also a tunable one available where you can pick the wavelength that you desire, ranging from 680 to 1050 nanometers. In addition to this, you can also do a dual modality system, the IVIM CMS or the IVM CM, which would combine the advantages of both the confocal and the 2-photon modality. So this is the footprint of the IVM system. As you can see, it is one of the most compact intravital microscopy platforms that is in the market right now. The system was designed with space efficiency in mind. We've endeavored to minimize its footprints without actually compromising its performance, and the result is that the microscope body is about 40 by 40 by 20 inches. And this compact design can be then placed in a laboratory space of your choosing and also enhances accessibility and convenience for researchers. So as you can see in this image, you can prop this microscope on a 1 by 1 meter table, and everything is enclosed and a compact footprint with the manual adjustments on the side. Another feature that is provided with this system is that it's a fully enclosed design. So access to the imaging chamber can be done through the front of the sliding door. And then there's also another access of a sliding door on the side in order to be able to monitor your mice. But once these two doors are closed, it's fully enclosed, and you don't require a dark room in order to do your imaging. And this is especially important for two-photon imaging. So in addition to the convenience of the size of the system, the IVIM system also comes with animal monitoring platforms fully integrated into the system. So one of these monitoring platforms is temperature. So if you see this image here, you have your XYZ translational stage. And this holds a plate heater that could heat up the animal that's under the imaging chamber. We also have a rectal probe that's in the animal that monitors the internal body temperature. And there is a feedback loop that then tells the plate heater to either warm up or cool down in order to maintain a stable body temperature. And this can all be viewed and adjusted on a tablet that would be outside of the scope. In addition to the plate heater, we also have a glass heater for the tissue that you're looking at. So you can have your imaging window chamber with a cover slip that's held on by a magnetic holder, and this is on a glass heater. And so you can image the organ that's right now outside of the body and keep the temperature stable. So this is another view of what the heating plate would look like in the imaging chamber. So the sliding plate holder is mounted on these rails that can slide in and out that allows for convenient access to the mice. The plate heater is then placed on the sliding holder, and it's held securely with 4 magnets. You can place your animal there with the rectal proof, and it could slide back into the imaging chamber in order to do your experiment. In addition to the plate heater here, we also have another version of the plate heater that is a stereotactic mount. This would be very beneficial for research that has to utilize cranial window imaging. And so you can see here we have a bite bar for the mouse, and then you also have the pincers that immobilizes the head. In addition, you have a variety of apparatuses that can help restrain the animal in a way that is convenient to you. And as you can see, these are highly adjustable and even removable according to your preference for your experiment. Another component for the animal monitoring platform is the LED light and camera that is in the imaging chamber. This allows you to directly observe the animal in between your experiments to make sure everything is physiologically stable. We also have integrated these gas inlets and outlets on the side of the microscope body for inhalation anesthesia if you're not interested in using injectables. So as you can see, you can actually connect your inlet and outlet hose that goes into the imaging chamber. And if you were to take a look inside, you have the appropriate connecting tubes and adapters that then go towards a customized nose cone for the mouse that allows you to use the anesthesia with ease. The IVIM system also offers many apparatuses that allows you to do long-term imaging for a variety of organs through these customized window chambers. This is done through a lot of research that has been done on how to properly expose the tissue but maintain its physiological conditions in order to do the experiment. And so as you can see for each application here, which is shown, it includes an imaging window chamber to optically expose the sample. It also includes an associated holder that would stabilize that window chamber onto the stage. And the kit would also provide with other consumables such as cover slips that would be required for surgical procedures. A lot of these kits also have corresponding protocols that have been created and optimized for each tissue applications. So here's an example of one of the window chambers that is popular. This is the abdominal window chamber. So as you can see, an incision was made on the animal's body in the abdomen area in order to expose the kidney, spleen, and pancreas. The window chamber was then inserted, and then the skin was sutured around such that it would tightly hold the ring in place with a cover slip on top. The chamber holder, which is attached to the stage, is then slid onto the ring in order to keep the imaging window stable for long-term imaging. And then this can be then used in order to visualize each of these organs. Here's another example for cardiac and lung imaging. With cardiac and lung, which has a lot of movement, this would require negative pressure mechanism, which is facilitated by a vacuum-based tissue motion stabilizer. This establishes a stable interface for a portion of the, in this case, lung tissue, and you ensure a secure and motion-free environment that then allows you to capture the dynamic behavior. So for example, in the image on the right, the video on the right, you see neutrophils and their dynamic behavior within the vasculature in the lung. As I said before, intravital microscopy requires ultra-fast imaging speeds in order to be able to capture fast dynamic movements, and this is achieved using a polygon scanner. So the polygon scanner stands as a technological cornerstone, and it facilitates high-speed in vivo imaging at an impressive rate of 100 frames per second at a 512 by 512 pixel resolution. What truly sets this scanning system apart is that it ensures that you can have uniform excitation across the entire field of view. The uniform illumination is very pivotal for obtaining reliable imaging data. You don't have reduced fluorescence signal to noise ratio at the center of the field of view, which is seen in other scanning systems. You don't have excessive photo bleaching at the edges of the field of view, and the illumination is seen to be uniform across the entire field of view, which improves the image quality without wasting excessive photons. And this guarantees captured images are not only rapid, but exceptionally clear from unwanted noise. Here are a couple of examples of how this polygon scanner helps to capture dynamics, fast dynamics within a system. So the video on the right is showing a lymph node, and you can see the T cells and B cells designated in purple and green, and their movement in the lymph node, which is highlighted by the blue channels. Another example is the inflammatory response in the skin. So here we are seeing an injury site in red, and you can see the granulocytes swarming behavior to the injury site, which would not be possible to visualize without a fast scanner. In addition to having a fast scanner, we also implemented a motion compensation feature. So this is a critical component for smooth imaging. This is a GPU-assisted parallel computing, and it allows you to acquire motion compensated images in real time, and it optimizes workflow efficiency. You're able to get these compensated by facilitating frame-by-frame motion compensation, depending on the channel you choose. So for example, in a cardiac image, you can choose whichever color channel that you know the structure should be stagnant, such as the vasculature. And using that channel, you can then do the frame-by-frame motion compensation. So just to show a comparison, this is a real-time intravital microscope video of the cardiac imaging with its heartbeat without motion compensation at a 15 frame average, you can see that the image without the compensation feature would be excessively blurry. But having the motion compensation feature, you are able to get very clear images then allow you to pursue dynamic imaging. As I stated before, we also have the two-photon modality that can be integrated into the system. Two-photon is a mechanism that involves the simultaneous absorption of two photons within a few femtoseconds, and this leads to fluorophore excitation and subsequent light emission. This is particularly valuable for live cell imaging, especially if you're looking at thicker tissues. And this surpasses the capabilities of confocal microscopy. In addition, you also will be able to do second harmonic generation microscopy, which is a label-free imaging modalities that can be detected in fiber structures such as collagen networks and these muscle fibers. So in this example, you can see the striated structure of a skeletal muscle. And you're able to visualize the neuromuscular junction and the vascular network. And this makes two-photon microscopy a powerful tool if you want to capture high-quality images for a diverse biological contexts. The IVIM system comes with a software engine, which makes the image acquisition to be very user-friendly, and therefore there's no need to have a dedicated operator for this system. The software allows users to pick their required lasers and detectors for their experiments. And it also allows you to easily adjust settings such as laser power, gain, and pseudocolor in order to obtain optimal signal-to-noise ratios. The software engine also allows you to automate imaging for 3D stacks, for tiling, monitoring multi-positions, and time-lapse imaging. So these four automatic imaging applications on the software engines can also be combined in order to maximize your data collection during your experiments. So for example, you could do a Z-stack and tiling at the same time, and the program would automatically adjust in order to acquire the data. In addition to the software engine, there's also the software studio that allows you to do image processing. In this software, you can do 3D rendering, maximum intensity projection, as well as time-lapse integration in order to visualize your data in 4 dimensions. The software comes with multiple options to save rotations and time-lapse videos as you see fit. So here we're showing a 2 rotations and a time lapse at the same time for the ear skin. And so this allows you to be able to visualize cross-sectional analysis, as well as look at cell movements and speed in a 3-dimensional way. So all these competitive advantages show that the IVIM system fully integrates everything you need in order to have a successful intravital imaging experiment. And this is a total solution provider for intravital microscopy. The accessories and expertise have shown that you can do this in almost any organ that you choose, as long as you can optically expose it to the objective. And you can see that they're very good signal to noise ratios, as well as having in vivo labeling kits allows you to see whatever cells and tissues that you require. And as stated before, a lot of research and innovations have been done in order to optimize these procedures and protocols, a lot of which have been published. So as you can see, they've been published in terms of their desired organs, as well as applications such as microcirculation, drug delivery, or cell transplantation, which you're welcome to check out.
Thank you, Dr. Karanendran, for the insightful presentation thus far. I'd like for us to pause for a moment and engage with our audience. We're curious to learn more about your specific interests in intravital microscopy. So our poll question, our second poll question for today is, what application would you be interested in looking at with intravital microscopy? This is a multiple choice question, so you can click as many answers that apply to you. Would it be immunology, cancer, cardiovascular, stem cell, neuroscience, regenerative medicine, lung imaging, or drug delivery? So remember, this is multiple choice, so you can click as many that apply. I'll give us a couple more seconds for you to choose before we can carry on. Great. Thank you all for answering. I'd like to give the floor back to Dr. Kiranandri.
Thank you, Ivy. So now we'll segue into intravital imaging in the context of preclinical research. As stated before, this could stand as a transformative tool in the landscape of preclinical research and offer researchers access to dynamic and the intricacies of living organisms at a cellular and molecular level. Unlike most traditional imaging modalities, you'll be able to do real-time visualization of biological processes. And this holds a promise for unraveling complexities of disease progression, immune responses, and therapeutic interventions in a physiological context. By providing a window into the inner workings of living systems, the intravital imaging opens new avenues to understand fundamental biological processes, as well as develop novel strategy for disease diagnostics, treatment, and prevention. This is an example of how intravital microscopy can be used in the field of neuroscience, as well as to understand the basic biology in a disease model. So we can visualize using the cranial window procedure. You can visualize the neurovascular unit, which is composed of pericytes, astrocytes, and endothelial cells in the cerebral cortex. And this would help us to analyze the integrity of the cerebral cortex, especially in the brain microvasculature integrity. You can also look at it in a disease model. So in this example here, you can look at the vasculature and leukocytes, as well as amyloid plaque accumulation that occurs over time. So this cranial imaging window, utilizing the stereotactic mount, offers a way to investigate dynamic cellular processes within the central nervous system in real time. And this allows you to visualize cell behaviors that would be incredibly insightful when looking at something like neuroinflammation or blood-brain barrier dysfunction. So here we're looking at a neuroinflammation model. And so, but the blood-brain barrier dysfunction is usually related to neuroinflammation in the central nervous system. And this has been implicated as one key factor in the pathogenesis of neurodegenerative diseases such as Alzheimer's and Huntington's. So here's an example of an animal model where neuroinflammatory in the brain is induced by injecting a chemical known as 3-NP. So after 3 injections, you have an induction of positive leukocytes, which are densely located within the cerebral cortex. And you utilize a transgenic mouse, which is the Lysem GFP. So this mouse model is it's been genetically modified to express GFP in a subset of leukocytes. We also additionally inject fluorescent blood tracers, so the dextran, in order to visualize the vasculature within the brain. And you can longitudinally observe the in vivo dynamic cellular behaviors and how this would affect the blood-brain barrier integrity in a 30-day neuroinflammatory state. Vascular leakages in the cerebral cortex, which reflect the impairment of the BBB, were observed at 2 weeks, which then persisted for the 3rd week. And by day 30, we were able to visualize severe inflammatory responses with massive leukocyte infiltration by day 30. You can also extend the study to the blood-brain barrier integrity. So we could look at immune cell dynamics within the blood-brain barrier, which is a critical interface that helps immune surveillance and for protecting the central nervous system. By visualizing the immune cell behavior in real time within an intact living organism, you're able to sort of unravel mechanisms that underline immune cell trafficking, infiltration, and activation in response to either neuroinflammation, neurological disorders, or injuries. So this precise and dynamic approach helps to enhance our understanding of immune-brain interactions, which was not possible with other more conventional imaging modalities. Here's another example using the abdominal window chamber. So intravital microscopy was used in order to conduct a longitudinal study of the abdominal organs, focusing on cell behavior in the pancreas and spleen. In order to achieve this, again, we would use a genetically modified model, the MIP-GFP mouse, which endogenously expresses GFP in pancreatic beta cells and allows you to track the dynamics over time. For the spleen, you would utilize another transgenic model that expresses GFP in monocytes and macrophages. And this enables you to observe these immune cells within the abdominal environment and gain insights to their role in health in a variety of diseases. This can be extended to the kidney as well, where you would visualize the behavior of transplanted islet cells and their interaction with the renal microenvironment in real time. You can track the dynamics of these islet cells and vessels post-transplantation, and it gives you insights on the mechanism of the underlying graft survival, the vascularization around it, as well as its immune response, which is information that's critical and improves outcomes in the field of regenerative medicine, as well as transplantation therapies. The system would also be able to do disease monitoring in cancer models. In this particular example, this is looking at the growth of pancreatic tumor and the vascularization around that growth. And so pancreatic cancer cells that also have a GFP are cultured and then injected into the pancreas. This is then followed by a window surgical implantation, which reveals the pancreas as well as the inoculation site. So you're able to visualize this over a number of days. And then you can compare between, in this example, day 9 and day 14 to see the growth of the pancreatic tumor, as well as see how the vascularization develops around the growth, as shown here in red. Because of the system's capability to 3D render and see the tissue in different orientations, you're also able to see the thickness of the tumor and the vascularization. So you can see that there's an increase in the thickness of the tumor from day 9 to day 14. This is just another view to show you can look at this day by day. You can look at the same site due to the imaging window chamber and actively track the development of this disease in real time. We can also use this real-time monitoring of disease progression in response to external factors such as diet, which is very important in the context of heart disease. So by visualizing cellular dynamics within the cardiovascular system, researchers can observe how dietary interventions can impact vascular health, plaque formation, and cardiac function over time, which offers insights to preventative and therapeutic strategies for managing heart disease. So here we're looking at the epicardial fat accumulation, which was induced by feeding the mouse a high-fat diet. So the white color that you see in the images is the accumulation of lipids, which was labeled using a particular fluorophore called SF44, which was then acquired in vivo imaging. Then you can see the fat deposition both in the superficial layer and the deeper tissue layer for 4 and 8 weeks. You can also use this technique in order to monitor cancer metastasis. So here we're looking at circulating cancer cells, or CTC, first in the vasculature of the lung. You can see in green the cancer cells that are localized within the vasculature, highlighted in red. You can see in the next image, which is in bone marrow, as shown with the stromal cell highlighted in purple, you can see the cancer cells circulating. And then finally, cancer metastasis in the lymph nodes, as shown in this particular image. So intravital microscopy allows you to have a higher resolution, but also monitor how cancer would metastasize within the animal. This modality also offers new insights in evaluating therapeutic interventions, which includes nanoparticle drug delivery in real time within living tissues. This would allow you to directly visualize drug distribution, cellular responses to the drug, as well as therapeutic efficacy, which is done in a minimally invasive approach. So in this example, you're looking at the liver using the abdominal window chamber, using a slight incision on the abdominal skin to optically expose the liver tissue and implant the window. You're able to see the drug delivery and where it is localized within the hepatic sinusoids and their subsequent accumulation over time. So this is a liver sinusoid shown in GFP and the drug delivery nanoparticles in red. This is another example of tracking nanoparticles in the context of cancer. Nanoparticles have emerged as promising carriers for delivering chemotherapy drugs. And so this can be used in order to determine the efficacy of the delivery system. In cancer models, usually the vasculature is disorganized, and therefore the drug can be stagnant and accumulate in places that are not required. And so with this particular imaging modality, you can actually visualize where the drug is distributed, where it localizes over a period of time. So here you're using the dorsal skin fold chamber for cancer xenograft imaging. So the dorsal skin is in between two plates that is then inoculated with cancer cells. And once the cancer has grown, you can inject intravenously the nanoparticle drug and visualize it within the imaging window over 24 hours. And so if I take the green channel away that shows the cancer cells, you can see in the vasculature where the red nanoparticles are then localized and accumulated over 24 hours. This can also be used in the liver if you want to look at drug delivery and clearance in the liver. So for example, if you're looking at indocyanine green, which is a biocompatible fluorophore used for determining liver function diagnostics, you can now do in vivo pharmacokinetic dynamic imaging in order to see how well it perfuses in the perisinusoidal space. So in the video here, you can see that it was intravenously injected, and you can actually visualize the delivery efficiency within the sinusoid of the liver. And then after a certain period of time, when there's no fluorescence, you can see the hepatic clearance of the compound. You can also look at the treatment efficacy in tumor vessels. So this is looking at tumor growth as well as the vascularization. So in the sham treatment model, you can see without the treatment, the vascularization becomes chaotic as the tumor grows. But with the anti-angiogenic treatment being administered in a different mouse model, you can see that despite the tumor growth, the vascularization stays a lot more uniform, which allows for a better flow and may also allow for better drug delivery. And so you can use this method to also check the treatment efficacy of these anti-angiogenic treatments. So in order to close today's webinar, I hope I was able to highlight the immense potential of intravital microscopy in revolutionizing preclinical research. Having the real-time high-resolution imaging of cellular processes within living organisms help to offer unparalleled insights into the complexity of the dynamic behavior in tissue structure. From tracking immune cell behavior to studying drug delivery mechanisms, the IVIM system helps researchers to unravel the mysteries of disease progression and therapeutic responses in a way that was previously not available. Thank you for joining us today on this webinar. And I will send it over to Ivy for the Q&A.
Thank you, Dr. Kiranendran, for such an interesting and insightful presentation. I'd like to open the floor to questions. And we have a question from the audience. Can we track the mRNA-based therapeutic compound as small as 20 to 50 nm in vivo, and how long can you image?
So with the Intravital system, we are able to do subcellular resolution, which allows you to look at subcellular organelles. So you should be able to look at mRNA-assisted drug deliveries. In terms of the time, that just depends on the model itself. So I would say maybe within 24 hours. But if the model is healthy and the drug delivery system is efficient, probably longer. But it just depends on the system.
Great, thank you. And one final question. How do I choose whether confocal or two photon modality would be suitable for my application?
Yeah, so both of these modalities offer different advantages. With two photon, you're able to dive into deeper layers of your tissue, and you're also able to do some label-free imaging. So if you require second harmonic imagings in order to look at collagen structure or muscle structure, um, it would be probably best to have the two photon modality. But if you would prefer to have a very wide arrangement of dyes, as well as have the optical sectioning, then confocal modality would be best. But with the IVIM system, you also have the option of having dual, having the best of both.
Great, thank you. So we've reached the end of our session today, and to be respectful of everyone's time, we're going to wrap things up. As mentioned at the start of our webinar, we will be sure to answer any outstanding questions in the written transcript, and we'll work to get this out to you over the next few days. I'd like to thank Dr. Karanandrian for this presentation, as well as for taking the time to address all of the questions you had. I trust that we have been able to provide you with some relevant information to the studies you were conducting back at your own labs. If in the next few days and weeks you have any further questions about the work that we've discussed today, or questions about any of our equipment, including the IVM system, I encourage you to reach out to us here at Syntica, and we would be happy to discuss further. We would like to welcome the opportunity to discuss your specific research goals and how many of our equipment could help move your work forward. Thanks again to all of you for taking the time out of your day to attend our session. We look forward to seeing you at our future Syntica events. Have a great day.